Hello students, welcome back to the channel. In this video, we are going to study how the production of monoclonal antibodies are done. So now see how the scientist will be doing it. First of all, a purified antigen is injected into the animal which is in the lab. Okay. Now animal is given sufficient time to develop the immune response and it will produce antibodies. Now, spleen will be harvested and lymphocyte, especially B cell, will be isolated. Okay, they are removed and they are cloned, which are producing a response to the antigen by making antibodies. Now, these plasma cells which produce antibodies, they are short-lived. Now you want them, they should be long-lasting and continue produce antibody. Then what we will do, we will try to fuse them with the myeloma cells. What are myeloma cells? These are cancerous cell. So this plasma B cell, which is producing antibody for a particular antigen, is fused with myeloma cell, and this result is known as hybridoma, which will have the characteristic of both cell, means it will produce continuously, it will produce cell, and because it is having characteristic of plasma cell, so it will produce antibodies, it will secrete specific type of antibodies. Is it clear? Now understand the diagram. So first of all, antibody forming cells you have isolated from the spleen. Now this is cell is fused with the tumor cell, means myeloma cell. This is myeloma cell. You have taken it from the culture medium. You have grown it there. And this cell fused is known as, now fusion is done. And finally, this cell is known as hybridoma. Clear? This is hybridoma. Now, this hybridoma, we will check it whether they are able to produce antibodies or not. And we will find they will produce antibodies only of one type. This is monoclonal antibody production. So this is summary here. Mouse is given the antigen exposure. Antibody formation is done. The B cell is isolated from the spleen. It is fused with the myeloma cells to form hybridoma. And then we will culture them and they will start producing antibodies, which is only of one type. We will say monoclonal antibodies. So I hope so in this short video, you might be able to understand how monoclonal antibody production is done. Thanks for watching.